Okay, so we are going to be covering topic 3b, 4, gametes, structure and function. The best way to revise for your test and your exams is to look at the specification. So what I'm going to do now, I'm going to read the points on the specification so that you know what things you have to concentrate on. So for example, it says 3.11 in the specification, understand how gametes in mammals are specialized for their functions. So you need to know about the sperm and the ovum. Mm-hmm. And specifically, notice they talk about the acrosome. What is the acrosome?
In the sperm.
Yes. What does it do? Because
it releases digestive enzymes.
It releases digestive enzymes, okay? So it's found in the sperm cell and it releases digestive enzymes. And the zona pellucida or pellucida in the egg cell. So they also talk about that. Tala, what is that?
It's the jelly-like coat and it hardens when there's, to like prevent many sperm cells from going in, so.
Okay, it hardens when?
When the sperm cell touches it, and then it only lets one through, I think.
Okay, so it only lets one sperm in at the time, one sperm nucleus in at the time. Why is that important? Tamsin, why is it important?
So like,
what would happen if more than one sperm could penetrate into the egg cell and take the DNA?
You'd have more than one.
It will have more than one set of chromosomes, okay? Remember, what is haploid and diploid? Holly, you remember? Haploid, diploid, we did that. Gametes are haploid or diploid?
Haploid.
Haploid. So gametes are haploid. This is something that you have to remember from meiosis.
Diploid is N and haploid is 2N.
No, haploid is N and diploid is 2N. Okay? And the diploid number of chromosomes is specific for each species. Mm-hmm. And in human beings, what is 2N? Holly, in humans?
23.
23 pairs. Mm-hmm. So the diploid number is 46 chromosomes. So they can organize them in pairs. That's why it's important. Because it comes from sexual reproduction. That means that one set of chromosomes is from the male parent and one set of chromosomes is from the female parents. This is biology and this, you cannot change that. So in sexual reproduction, you have the two gametes that were formed by meiosis, so you got the haploid number. And then you have the gametes are specialized. When we talk about a specialized cell, we need to remember that we have to talk about features in that cell that will allow it to carry out the function. So what is the function of gametes? Is to bring the DNA from the parents that are going to combine into this new individual. Okay? So for this test, all the topics that you have to do with reproduction, development. Okay? So these are the important things that you have to remember. So there will be questions about the acrosome and the zona pellucida, but there are also other structures or other, what we call in biology, adaptations in the gametes that are important. Can you remember one organelle that is found in abundance in the sperm cell and why it's so important? One cell organelle that is really highlighted when you talk about the sperm cell.
It's not important.
That it is. That is found in abundance in the sperm cells. And it's the mitochondria. Very good. So why is it important that the sperm cell contains a lot of mitochondria?
It provides a lot of energy, so it allows the tail to propel and move.
Okay, so it provides a lot of energy through aerobic respiration that propels the tail. Okay, very good. You always need to look also at the number of marks. I'm making just questions that come to my mind from looking at the specification. There are key points that you can have on your test or on your exams. Okay, So this thing with the mitochondria in the sperm cell, it's important. The zona pellucida, you need to be aware that it's this jelly-like that surrounds the egg. And it's important because it ensures that the diploid number is going to remain in that embryo that is going to be formed. So what is the name of the cell that is formed right after fertilization?
Zygote.
The zygote, very good. So the zygote is formed once fertilization takes place. And how can you define fertilization, Tansy?
Like the steps?
No, what is fertilization?
When the sperm cell reaches the ovule and it, like, I don't know how to explain it.
And the nucleus of the sperm fuses with the nucleus of the egg. And then the pairs are complete. Okay? So the diploid number is restored and you have the formation of the zygote. Then what happens with the zygote? What kind of cell division, Holly, happens in the zygote so for it to become an embryo?
Mitosis. And what was the question?
Yes, what type of cell division happens when the zygote is formed in order to become an embryo? So the zygote is there.
Mm-hmm.
There has to be cell division for the zygote to become embryo.
Is it not meiosis?
Meiosis.
Yeah.
So you all agree it's meiosis.
Mm-hmm.
What is meiosis?
It's, there's two nuclear divisions and it's only for sexual reproduction.
So once you have a zygote, are you going to have meiosis?
Fertilization, right?
Meiosis produces the gametes, okay?
Yeah.
The gametes got together through fertilization and form the zygote.
Yeah.
Now the zygote has to grow into an embryo.
Mm-hmm.
So is it going to be meiosis again?
Mitosis.
It's going to be mitosis,
Oh, yeah.
okay?
It's embryo development.
Because mitosis is a kind of cell division that produces what? Every division, there's going to be a question on your test that you're going to do later, so that's why I'm guiding you.
Mm-hmm. Produces.
So mitosis produces what?
Two daughter cells.
Two daughter cells that are going to be
One, like a zygote.
between them. What are they going to be? Alike, different, half? What's going to happen?
Different.
Identical.
Oh,
Very
yeah.
good. Yes. Okay. So you see, sometimes you have to put things into perspective.
Yeah.
So once fertilization takes place, the zygote is formed. For it to become an embryo, growth needs to happen. The kind of cell division that is involved in growth is mitosis. Why? Because for each mitotic division, one parent cell is going to divide into two identical daughter cells. However, when we have an embryo and the embryo eventually becomes a fetus, for example, a human fetus, there is going to be differentiation of cells, and that is called development. And that is also going to be on your test, and that's the next part of it. Okay? So that's when we need to talk about some other definitions. Okay, so we talked about fertilization, the acrosome reaction. You have to know it in detail. What do you remember about the acrosome reaction? There are some steps here on page...
It gets triggered when the sperm touches the zona pellucida.
Okay, so the sperm reaches the zona pellucida.
And the acrosome reaction is triggered.
And what is that about?
I don't know.
Holly, you said what the acrosome does.
It releases
It releases digestive enzymes. And what does those digestive enzymes break down? Because digestion is about breaking down larger molecules to small molecules.
They break down the zone.
The zona pellucida. Okay, so that the nucleus of the sperm can reach the nucleus of the egg.
Mm-hmm.
Okay. What is the difference between fertilization in mammals and fertilization in plants?
And one is
Plants
asexual.
Plants is what? Plants is what, Holly?
It's double fertilization.
It's double fertilization. Why is it called double fertilization? What is double about it?
Got two nuclei.
Mm-hmm. Which of the gametes, the male or the female?
Female.
Okay. In the female, there are like 8 haploid nuclei.
Yeah.
Okay, so it's quite a lot. But what is the name of the male gamete in plants?
Pollen grain.
Pollen grain. Okay, so the pollen grain is the male gamete in plant. And what's the name of the female gamete, or where is the female gamete found?
Is it not in the
ovule?
In the ovule, which is found inside the ovary. Okay, they also have ovaries. So the ovule. And the ovule will eventually become, from IGCSE?
Seed.
The seed, very good, with a baby plant in it. Okay, because we are looking at it again as formation of new individuals. It's interesting to remember that the ovule will become the seed, and inside of it is going to be a tiny plant, okay, that is a result of double fertilization. Now, the gamete that has the 2 nuclei is the male gamete. So the pollen grain will have the 2 nuclei. And then they will go down the pollen tube that is formed. Okay, so you need to know those steps as well. And then there will be a fusion of one of those nucleus with the egg nucleus, and that will form the embryo. And what is the other male haploid nucleus going to do? It's going to join what and form what? It's something a bit strange there, or particular in plants. Holly, we did that the last lesson we had. Remember? Here. So this is the embryo sac. You have the 2 polar nuclei. So you see, it's all N, so it's all haploid, and it's 8 of them. So you have 8 haploid nucleus. This is just the inside the ovule. Okay, and there are going to be these 2 nucleus. Look, this is a pollen grain, and it has 2 nucleus, 2 haploid nucleus, and they are the ones that are going to go down the. It's here. Down the pollen tube, and it's going to reach the embryo sac, which is inside the ovule, you see? And then, so you need to know this as well. One of the nucleus is going to fuse with the female gamete, and then the other one, the second male gamete, fuses with the polar nuclei, and it's going to produce a triploid. It's going to produce a triploid endosperm nucleus. It has 3N. And the function of the endosperm is just going to, it's going to have all the nutrients for the little embryo to grow during germination. Okay? Right, so that's double fertilization. Did you do this part, treating infertility?
No.
Okay, so we're going to skip this. This is the bigger part. Okay, there are some questions now. You can answer this one with the sperm cell, the one you have in front of you. Also, this other question is probably not going to apply. only the part about the sperm cells.
Like this.
So do 7A1 and 2. And you can do 7C as well.
This question, it was like in our test exactly this. So was these. Like our quiz that we did. So I don't think it will be in this one.
Yes, that's why I thought.
The exact one that we did.
Exactly.
Yes.
Yeah, because you told me meiosis was in, so I thought I would make that copy.
Yeah.
But now that we start from here, it's not going to be. So you're going to have the sperm definitely, because it's the same year, the 19 paper.
It's the exact same one that we had.
But I told you where to look.
I know.
Is it?
Yeah,
All of it?
I thought the whole thing. That was in our test.
That was too.
That was in our quiz.
Yeah.
She's definitely going to get us like one of these. She's definitely going to get it.
So let's tell me when you are done with the 7A and C. I didn't make a copy of B, but now that I'm thinking about it, maybe we should have a look at it. And C, right? What does C say? No. And we'll do D after we explain stem cells.
Yeah, forgot the cortical reaction.
Yeah. What's the cortical reaction again? Is that when it reaches it, then the cortical granules get released or something? I can't remember the cortical reaction.
Okay, you can look it up because you're studying. You need to know it. Or in your notes. Let me have a look at your notes because I haven't seen these notes.
So these are probably going to be on our test.
Yeah, for sure.
Probably.
Yeah.
But also you have some good material here in the book because these ones are not in your past papers. So like these questions, you can take also questions from here.
She gave an exam question. Describe and explain the way that the Acrosome reaction in humans prevents more than one sperm from fertilizing the egg cell. Like whenever she gives these, she
Puts them in the test.
Yeah, but that's what you described.
Mm-hmm.
Where is the other? Where is 3B? You have it?
Yeah.
Ah, okay. You're using it? All right.
All I know about the cortical granules, not about the reaction. Because the granules, the enzymes which release chemicals that cause the zone of Pelagia to harden when the nucleus of the sperm enters the egg cell.
Mm-hmm.
Is that it?
It has to do with that. How many marks is it? You always have to look at the number of marks. Yeah. And read the question carefully. What does the question say? Ask exactly.
Describe the role of the cortical reaction in the process of fertilization.
The role, you see? Describe the role in the process of fertilization. So we discussed it a while ago.
Yeah.
So think about that. I'm not going to tell you. I want you to write and then we discuss it.
Mm-hmm.
So what is the role of the cortical reaction in fertilization? You see, here in the specification it says Acrosome reaction So the question could have been, what is the role of the acrosome reaction? And that's what Holly explained with the enzymes.
Yeah.
Another possibility, the cortical reaction and the fusion of the nuclei. So those are three things. There are three different things, and they can ask you different questions about it. I touch the screen as if it's going to do something. Does it happen to you too?
Yeah.
You need to know fertilization in flowering plants. So there could be questions on that. Role of mitosis and the cell cycle in producing genetically identical daughters. Okay, we did that. What's the next part? Okay. Oh, it's a bit different in the book, the order. Because DNA methylates that there, then it goes here. You didn't do any of this?
No.
No, right? Not yet. Because she's following the order in the specification. So then it goes here. Okay. Okay, let's see. Who wants to start?
We all wrote the same thing, I think.
Okay, let's see. What's the question? Tala, read the question, please.
Explain the role of the cortical reaction in the process of fertilization in mammals.
Okay. Holly, what did you write? How many marks?
It's 3.
3 marks. Okay, let's see.
I said the cortical granules are released from the egg cell, causing the zona pellucida to harden, creating the fertilization membrane that stops any other sperm cells from penetrating the egg cell, preventing polysperm.
Okay, so for writing the zona pellucida hardens, you get 1 mark. So if you put that, you get 1 mark. Or you could have put as well that it becomes impermeable, but you use the correct term. Mm-hmm. The role is to prevent more than 1 sperm nucleus entering the egg cell, or you could have written to ensure that the nucleus is diploid. Mm-hmm. Okay, it's the same thing. So you get another mark for that. Holly, you mentioned something about cortical granules.
Yeah.
So here another mark for writing that cortical granules fuse with the egg membrane and releases enzymes.
So what part of that was right?
The hardening of the zona pellucida stops any other sperm cells from penetrating the egg.
And do your artwork to ensure the nucleus of the sperm is diploid.
Those are the 3 points. So one is the zona pellucida hardens, one is the cortical granules, okay, that secrete the enzymes that cause the hardening, and their role is to prevent more than 1 nucleus or more than 1 sperm going in and fusing with the nucleus of the egg to ensure the diploid number remains, okay, so that it can be a viable zygote. Because you know what would happen if you have another nucleus of another sperm entering, would be that the zygote will not be viable, meaning that it will die, it will not grow. Okay, it's an anomaly. That's normally what happens. Okay, that's 7C. What did you write 7A? 7A shows you a sperm cell, and it points to several parts. One part is the nucleus, the acrosome, the flagellum, and it's asking you, what is the question? Tamsin, please read it.
How many of the labeled structures contain DNA?
So how many of the labeled structures in a sperm cell contain DNA? What did you write, Tamsin?
I put A.
A, which is 1. Tala, you?
I put 1 as well.
And Holly? 1. Okay.
It's just the nucleus.
Yeah.
Yes. The nucleus is, because the sperm is one cell, the nucleus contains DNA, but the correct answer is not A.
Oh. We all took it.
Okay. So it's a very tricky way of asking or checking that you will remember that there is another organelle present in the cell that also contains DNA. Can you remember? Is there another? No, this is from the previous chapter. When you did prokaryotes and eukaryotes and you did the organelles of the eukaryotic cell and you did mitochondria and chloroplasts.
So
Hint. So mitochondria what? Contain DNA?
Yeah.
Mitochondria also contain DNA. So it's a very tricky way of asking that. Yeah, so the answer is 2. So it's B.
You get that's close.
Very tricky. Okay. 7A2. Holly, what's the question?
Sperm cells contain mitochondria. Describe the function of mitochondria in the movement of sperm cells.
And it's 2 marks. What did you write? So describe the role of mitochondria in sperm cells.
I said it gives energy to the sperm so it can move to reach the egg cell by aerobic respiration.
Very good. So you got the 2 marks because you mentioned aerobic respiration and you mentioned it provides energy. Okay? The moving part, because you didn't mention what specific part moves, it wouldn't give you the mark. You have to say moves the flagellum, but because you wrote it was only 2 marks, you got it. Remember in these questions, because it's now A level, you need to show that you know all the details that you're supposed to know. So like now we're doing reproduction, but they're still asking you questions about organelles in eukaryotic cells. Okay? Right, so we did 7A, 7C, 7B. I didn't show it to you, but let's have a look. Because who knows, you might have it. Oh, what did I do? No. Restore. Oh no, I haven't. Why do I do that?
Check your history.
How do I do that?
Go to your profile and then settings.
Here?
Yeah, there's like a few dots, I think.
These ones?
No.
These ones here? Yeah.
Then there's history.
Oh, yes. I knew there was an easier way.
And it's
recently closed. Perfect. It doesn't give me the option, but it has 11 tabs. Here, yeah. They're here. Past papers. This one. No. Right, okay.
Well, you can drop your guys home.
The keys to my husband texted too.
Holly, are you tired? You look tired today.
Yeah, I'm so tired.
Did you have more tests?
Yeah, we had a test today.
Of what?
ICT.
I had 2.
It's that time of year.
Yeah, in February they just give you so much.
No tests.
You have 2 tests next week as well. Yeah, that's 2. What was on the week?
There. So it's question 7.
Because I've done 5 anymore.
Because I wasn't in for last week.
What page does it say? For the sperm thing?
I think it's like 33 or 22.
Yeah.
Okay. Ah, there it is, this one. Okay. Can you see it? I can make it bigger. Okay, so this is a Chinese hamster. This is 7B. Okay? Because I have the... No, but because I thought we were going to spend more time on the other one. So can you all see?
Yeah.
Okay. So it says the photograph shows a Chinese hamster, magnification times 1, which means this is the actual size. Right, then female Chinese hamster mates with many males in a short period of time. The diagram shows the relative size of a Chinese hamster sperm compared with a human sperm cell. So it shows a sperm cell with a very, very long flagellum. Okay, then suggest why the Chinese hamster sperm cell has such a long flagellum. I think this is 4 marks. It's quite a lot. So, what can you think of?
There's more.
3 marks.
I mean, there's more obstacles. Like, not obstacles, but. He's in a longer way to go. Like, more. I don't know how to explain.
Actually, no, the human sperm has a longer way to go because, I mean, the hamster is tiny.
Oh, yeah, true.
But what it's telling you is that one female hamster mates with many male hamsters in a short period of time. So, what is the obstacle? What would be the correct word to use so you will get a mark?
The other sperm cells? The different sperm.
So, what happens between different sperm cells?
Genetic variation.
Keyword. What are the sperm cells doing between them?
They're like
Competing. So there is competition between sperm cells of different male hamsters. So the long flagellum will help with what?
The propelling.
Like fighting them all.
To get there first.
2 marks. No, you have the idea.
Yeah.
I'm just helping you to get to the keyword that is going to give you the mark. Okay? Because yes, it is a fight, it's a struggle, but the correct word in biology for that is competition. So the sperms have a lot of competition because there are a lot of sperms. So the longer flagellum will help it get to the egg cell faster. Okay? That's it. Okay?
So do humans. They have like millions of sperm cells.
Yes, and there will be the one that will manage to do the acrosome reaction is the only one that is going to actually go through. And then that sperm probably has a longer flagellum than the others, or has more enzymes, or has more mitochondria, or, is best suited, and that's all evolution. Because that's the survival of the fittest. Okay, this is at the cellular level. Let's check if we missed anything from the mark scheme. So history. I learned something today. I think it's this one.
If this comes up on the test tomorrow, I'm going to be jumping through.
Well, there's a high possibility.
That's like 10 marks
Did I
in
get
the
well?
bag. That's like 12 marks just done.
But you can do this on your own every time you have a test. You know that.
I never know where to search.
What's it on? She said it starts Pearson.
It's you go to International A-level IAL Biology Pearson qualifications, and then you go to biology, and then you find the 2018 specification, because the papers you have are older. They're from the previous specification. Okay. And then you find the mark scheme right next to it. Okay, so for B, the sperm with the longer flagellum swims faster. So this is what Holly said, 1 mark. The sperm have increased chance of fertilizing the egg cell. Oh, so, okay, this was the mark that we didn't think of.
Sorry,
It's okay. No, just going back to the hamster. Okay, the one that we didn't mention was that the sperm with the longer flagellum has a higher chance of fertilizing the egg. Which again, it's like it's given, but if you don't write it down, then you don't give them out. It's like it's so obvious sometimes, some of the things. And then competition with sperm from other male Chinese hamsters. So those are 3. Now, other things that you could have thought of, because they give us different options here, because it's a suggest question, which are the hardest ones. You could have said a longer flagellum gives more force. Could have thought like that. They accept to be the first to fertilize the egg. But look, ignore if you write is the first to reach the egg, you don't get the mark. But if you say is the first to fertilize the egg, then so that's why you have to be very careful when you answer in biology, always to use keywords. Okay? And then for the competition part, accept the description of competition with sperm from other male Chinese hamster. Also, you could have said it's a consequence of evolution, which was what I told you. It's about evolution. Okay, survival of the fittest. So it's important to pay attention to this, because they will always ask you something that they will expect you to mention these things. Okay? Right. Okay, we did 7C. Now let's look at the other terms from the specification here. History again. So 4. Do you have any questions from up to here? Okay. History, and then the tabs, and then where is it? Excel, this one. Excel International Advanced Level Biology. Person qualifications is looks like this. This is the specification. So that's where you have. But I had my PDF. Where is it? Is it here? No. Oh, there it is. Okay. Okay, so when you're going to study for your final exams, you're going to use this. Believe me, it's going to help you a lot. Okay, we were somewhere here. So now, you need to know the following terms. Stem cell, pluripotent and totipotent, morula, and blastocyst. Okay, so you need to define all of those terms, and then be able to discuss the ways in which society uses scientific knowledge to make decisions about the use of stem cells, but you said you didn't do that part.
Yeah, we did. We did.
Okay.
The regulatory authorities.
Okay, and medical therapies. And then I think this part is also in understand how cells become specialized through differential gene expression, producing active messenger RNA, leading to the synthesis of proteins which in turn control cell processes or determine cell structure in animals and plants. This is something that is probably not written as such in the book, but again, it's like a consequence that you are expected to know. Okay? Shall I bring your coffee, Holly? Green tea?
No, yeah.
Something to wake you up? A Coke? Or you can stand up and stretch and move.
Don't worry.
Okay. So understand how one gene can give rise to more than one protein through post-transcriptional changes. Did you do that as well?
I
Yeah,
don't get it. She says it and she says rice. Is it rice? Like
Rice?
Is it life or rice? Because whenever I hear her say it, I'm always thinking about her saying rice.
For what? For
giving to something. So
giving rice. Rice, yeah, but with an S.
Oh.
Yeah.
I'm always thinking about it when she says it. I'm like, is she saying rice?
Oh,
We do
okay.
have post-transcriptional though.
Okay, so that is in. Understand how the phenotype is a result of interaction between genes and the environment, epigenetic modification. You said that bit is not in, so that's this bit here. It's quite a lot of material actually.
Yeah.
We need to. We have a question in 7. Question about that. Okay, so understand how the phenotype is the result of the interaction between genotype and the environment. Know how epigenetic modification, including DNA methylation, did you do that? DNA methylation and histone modification will alter the activation of a gene. Let me see.
Histones, yeah, I don't understand what histones are.
This one.
Like all the chemistry not part of this.
Histones are the proteins around which DNA is coiled in the chromosome. And they play an important role because when the DNA is tightly coiled around the histones, it cannot be transcribed. So you have to make the connection now with what you learned when you did protein synthesis. Yeah, you did it. Is it all in? All of this? Yeah, you see histones, so the coil of the DNA in the chromosome. And the histone, so for the DNA part to be accessible to the enzymes that are going to what is produced during transcription?
Protein. A single different protein.
No protein yet. No. Transcription happens in eukaryotic cells, it happens in the nucleus. So what is produced during transcription? You have to remember, because otherwise you're not going to understand this part. What is produced during transcription? mRNA. Genius girl today.
I thought it was wrong.
Yes, very good.
She said it.
Who said it?
No.
Okay. Very good. Yeah, so that's what you need to bear in mind. So that's the role of histones, because you're asking what is the connection now with histones. If the DNA is tightly coiled around the histones, while it's coiled, it cannot be read by the enzymes that will produce messenger RNA. If it cannot be transcribed, then it cannot be expressed, because a gene is expressed when a protein is synthesized. When the protein that is coded for, that's the term you have to use. Proteins are coded for by genes, and genes are sections of DNA. Around 5,000 nucleotides, okay, or more, make one gene. That gene has the information to code for a polypeptide chain that is going to form a protein. So when the DNA is tightly coiled around the histone, it cannot be transcribed. Therefore, it cannot be expressed. So these are the keywords. Transcription, expression of genes. Expression of genes is going to be after translation on the ribosome, when the protein is produced. Okay? So that's why in this process, because it's not a simple process, it's a rather complicated process. And that's how genes are switched on and off. So you want the bad genes, let's say, to be switched off, and you want the good genes, the genes that are going to produce proteins that are going to make us healthy, that are going to make us alert, that are going to make us even happy, the hormones. You want those genes to be switched on. Okay? And you can do that through all of these processes that are mentioned there. Methylation, epigenetics. That's what epigenetics is about. Epigenetics is about using the fact that yes, we have these genes, but our lifestyle can determine that these genes will be switched. We will switch on the good ones and we will switch off the bad ones. Okay? Now, we don't really use the terms good ones and bad ones, but it's to give you an idea. Okay? Because the genes code for proteins, and proteins can be enzymes or can be hormones or can be enzymes that determine the presence of certain hormones. And we want the hormones that give us well-being. So that's the importance of epigenetics, and that's why it's so out there nowadays. And this process of methylation, what it does, we will see it in a minute. And the other thing you need to understand is after the genes are transcribed and the messenger RNA, before it leaves the nucleus, this splicing, what is splicing? This is another keyword.
RNA splicing.
This is splicing, just in case you remember.
I wrote it down.
We did that with mine today, didn't we? Remember when she was doing it?
Can I read it?
Yeah, read it. Yeah, find it. It's important. Splicing is a very important process.
It's the organism, when the organism can produce more proteins than the number of genes available.
Okay, that's not exactly splicing. That's alternative splicing. Splicing on by itself, it's cutting the DNA, in this case the RNA. So you see the DNA, here you have to start DNA, arrow RNA, that's transcription. Okay? Then the RNA has to be modified before it leaves the nucleus. And splicing is just cutting the RNA in certain areas. And that is done by enzymes. But there are sequences of bases that will determine where it's going to be cut. And what are they going to be cutting? They're going to be cutting the sections of bases. Remember, these are all bases. So A, U, U, T, G, not T, G, C, whatever, sequence. Okay? There will be some parts that don't code for anything. And they need to be removed from the RNA before it leaves the nucleus. That is called splicing. Now, when you have the RNA section and it's chopped, then alternative splicing means that when the segments are going to be put together again before they leave the nucleus, they can be put together in any order. So if we call them 1, 2, 3, 4, 5, they can reconnect in the same order without the introns, which are the bits that do not code for anything. We want the exons only. So there are exons and introns. Okay? The introns is like, well, you are there, you are doing nothing, we have to remove you. When the exons are going to get together again, they can get together in different sequences. So not only 1, 2, 3, 4, 5, but 1, 3, 4, 2 in different ways. And that creates variation. So you can get different polypeptide chains. So this will be translation. So from messenger RNA in the ribosome, you get the polypeptide chain. And because the order has changed, then you have even more options for different polypeptide chains. Okay? So splicing is cutting, removing the introns. Okay? And alternative splicing is the different ways in which the exons are going to get together, okay, before translation. Okay? Any questions about this? You need to know this. Okay.
Yes, can I use your bag?
Yes, go there. You know where it is, right? Next to the door.
Yeah, I think so.
The door is open, so you will see it. Okay. Now, why is this important? You want to have a little break?
Mm-hmm.
Sure?
Yeah.
Okay. More water?
Alright.
Fine, thanks.
Okay. So now, shall we follow this? Is this a good idea? Do you?
Yeah.
Okay, let's go over the terminology. I like to use the book, always. You have that, and I will use the book. So, I'm using stems.
She also gave us this revision, and I don't know if the answers are right because I haven't checked them, but she gave us this, and she said questions 1, 3, 4, and 5 were important for the stem cell chapter.
Okay. Yeah, because I have all these definitions. Yeah, this one. Double fertilization. Okay, we will have a look. But let's start from the definition. So, what is a stem cell? What's a morula? What is a blastocyst? Let's see. It says here, fertilization. You can't see with glasses. I can't see without glasses. Okay, fertilization starts a complex series of events. So when the zygote is formed, as I told you, mitosis starts. Mitosis is important for growth. And then you end up with a ball of cells. Okay, so here it says the very earliest cells in an embryo are totipotent, like the zygote. So you need to know what that means. What is totipotent? Any ideas?
I remember speaking about it, but I don't remember what
I'll give you an important piece of information. There are 216 different types of cells in humans. Okay?
They can form any one of them.
So a totipotent cell is one that has the ability to form any one of the 216 different cell types. Toti from Latin tutto, if you know Italian, is all of them. Potent has the potential to become all of them. So totality, you can think of it like that.
How many are there?
216
You have to know
in
the
humans.
specific number.
No, I'm just giving you the piece of information to illustrate what is a totipotent cell. So it's in the zygote, right? And then all of them are divided by mitosis, so you need a basic number of cells before they start differentiating. Okay, then the future role of individual cells are decided quite early in the life of an embryo. So you need to think of the ball of cells. You need to think that as it grows, one end of that ball of cells will become the head and the other end will become the limbs. Okay? So from there, already differentiation starts. Differentiation implies specialization of the cells. And that's what development is about. So the first stage of embryonic development is known as cleavage. Cleavage involves a special kind of mitosis, where cells divide repeatedly without the normal interface for growth between divisions. So there is no interface. It's just very, very fast division. The result of cleavage is a mass of small identical and undifferentiated cells that form a hollow sphere known as the blastocyst. Okay, cyst, again in Latin, means a ball. Okay, blastocyst is a hollow ball of cells, so there is nothing inside, and it looks like this. Okay, this is a 5-day blastocyst after fertilization. So in humans, this takes between 5 and 6 days. So if you remember that, you can write that if they ask you what a blastocyst is. Takes place as the zygote is moved along the oviduct towards the uterus. So remember, fertilization happens in the fallopian tube, and then this ball of cells is going to be moving until it's going to implant in the uterus. So as it moves, it develops. Okay? So the blastocyst has undifferentiated cells. Are they totipotent?
No.
Yes.
Is it?
They are totipotent.
Okay.
Okay? Because they are undifferentiated. Okay? So any undifferentiated cell is totipotent. So we will see. Okay, one large zygote cell forms many small cells in the early embryo. The tiny cells are also known as embryonic stem cells. Stem cells are undifferentiated cells that have the potential to develop into many different types of specialized cells. So they are totipotent. Okay? The very earliest cells in an embryo are totipotent. It's written here. By around the 4th day after fertilization, they become a solid ball of between 10 to 30 cells, known as the morula. So what's a blastocyst? It's a hollow ball of cells, undifferentiated and totipotent. What's the morula? Now it's more specific. You need to know the morula is a solid ball of 10 to 30 cells. Each of these cells in the morula is still totipotent and has the potential to form every type of adult human cell. Okay, we're still in totipotent and we are 4 days after fertilization. Within 1 more day, the cells have divided more and they have formed the blastocyst. So first is the morula and then is the blastocyst. By the time the blastocyst is formed, the cells in the inner layer have already lost some of the ability to differentiate. And the outer layer goes to form the placenta. Okay? The inner layer of cells can form almost all of the cell layers. Layer of cells. The inner layer of cells can form almost all of the cells needed in the future, but not tissues such as the placenta. So now they are not totipotent anymore. They are now pluripotent. Okay? So let's recap. Zygote. Then after the zygote, what is the next structure? The ball of cells.
Plus.
No, okay, now we are going into more detail. Okay? Morula. So zygote is one cell, then ball of cells, morula. Totipotent? Yes or no? Yes. After morula?
The blastocyst.
Okay, blastocyst. Now, the blastocyst, something happens there. At the blastocyst stage, the placenta is going to be formed by the outer cells of the blastocyst, which is a ball of cells, but it's hollow inside. So the cells that are inside cannot become placenta cells. So they are not totipotent anymore. What are they? Pluripotent. Okay? So again, Holly, zygote becomes morula. Morula becomes? Okay. Now Tala, the morula, totipotent or pluripotent cells? Totipotent. Tamsin, what happens during blastocyst stage?
Becomes pluripotent.
Which ones?
The
The inner cells of the blastocyst
placenta.
become pluripotent. The outer ones are going to form the placenta. Remember, the placenta is a structure that attaches the embryo to the mother, to the uterus wall. Okay? So, at the blastocyst stage, the outer cells are going to become the placenta. The inner cells are going to become pluripotent. They lost the totipotent capacity because they are not able to form placental cells. I think if you know this, you are in. Okay. Okay. Now, the genetic reason why the cells inside the blastocyst become pluripotent, in case you're asked, is because some of the genes have already been permanently switched off. So now it is important to understand how you switch on and off genes, because this is what gives the cells the ability to become specialized. When I say to become specialized, I mean that one stem cell is going to become a neuron, another stem cell is going to become a stomach cell, another is going to become a bone cell. That's what we mean when we say specialized. And in order to do that, different proteins have to be produced, and remember, the proteins are produced by the genes. So certain genes will be switched on in certain cells found in certain area of the blastocyst. Okay? Right. Now, types of stem cells. Here you have embryonic cells, umbilical cells, and adult stem cells. So there are 3 types of stem cells. The embryonic cells are the ones that we've talked about, the pluripotent that are in the blastocyst. Okay, they change to become more specialized as the embryo develops. So let's read this bit here. For example, they might form blood stem cells, which give rise to blood cells. The blood stem cells are found in the bone marrow. Okay. By around 3 months of pregnancy, the cells have become sufficiently specialized that when they divide, they form only more of the same type. What does that mean? Muscle cells will divide by mitosis and form more muscle. So the baby now, after 3 months, the fetus, call it the embryo, start calling it the fetus when all of the organs are already formed. Okay, and it's just growing. And growth happens through mitosis. Now, there are some stem cells that are found in the umbilical cord, and now they have this. I remember 12 years ago when I had my youngest child, they were offering umbilical cord. What was it? It was like a storage. You could store it because if the child in the future has, for example, leukemia, they can use those stem cells to.
So the body won't reject it.
So the body won't reject it, and because they are pluripotent. This is what you need to know for your test. It says here, the blood that drains from the placenta and the umbilical cord after birth is a rich source of pluripotent stem cells. Okay. Okay. Adult stem cells. So in adults, where do we find stem cells? Do you have any ideas? Do you remember from the lesson?
Where do you find stem cells?
No.
Bone marrow.
Bone marrow, yeah. So white bone marrow contains stem cells. They can form white blood cells. There are only a very small number of adult stem cells in each different tissue. So can you think of another tissue where there are stem cells, where there is a lot of mitosis happening and
specific tissue?
We have so many tissues in our body.
The heart. The heart.
The heart is one of those organs that
Muscles.
Yeah.
muscles if you work out, but another one that even if you don't work out and you don't eat a lot of proteins and you're not into that, your skin. Okay, your skin cells. It's under the dermis. They're always dividing, so you could find stem cells there before they become actual skin, because they have the ability to.
If there's a burn victim, then do they use this stem cells to make new skin?
Yeah. It's amazing now for treating burns, second-degree burns. They have these. It's like covers. They cover you.
Yeah, I've seen it.
Yeah, and it's like a gauze, and it has stem cells, and then they just go there and they just become like the skin of the person. It's amazing. Okay. So that's about the stem cells. Then the development of the organism. Now here you need to talk about this thing with the genes being switched on. So a totipotent cell. So now pluripotent is off after blastocyst. So remember, pluripotent finishes with the blastocyst.
What does pluripotent, like, what is it? Because
it's like most of the cells are
most of them.
Yes, pluri means many. Many except one, which is the placenta.
Okay.
Toti is the totality of them. Pluri is many.
I read apart from the blastocyst. So is that the same thing then?
What?
The definition of pluripotent is like most of them apart from the blastocyst.
In the morula. Up to the morula, then it's totipotent.
Yeah, show you.
From blastocyst onwards, it's totipotent. Is that what you're saying?
No, let me show you.
Okay.
Pluripotent stem cells, undifferentiated cells that can form most of the cell types needed for an entire new organism, apart from the cells of blastocyst.
Apart from the cells?
Of blastocyst.
Okay, yeah, it's a complicated way of saying the same thing, because in the blastocyst has the ability to form the placenta.
Oh, okay.
That's why. Okay.
It's confusing.
But you get it, right?
Yes.
So toti is everything including the placenta, which is up to the blastocyst stage. From the blastocyst onwards, when the embryo grows, and eventually will form the fetus, then all the other, let's say, tissues and organs will be formed. Except the placenta, because the placenta is just there. It was from the outer cells of the blastocyst. That's why it says except the blastocyst. Okay? So, totipotent up to blastocyst. Now here it says switching on and off of genes. Here, we are on page 210. Okay, so it's revision, so you have to remember the things you studied before during protein synthesis. So what do you remember about switching genes on and off?
They're activated and inactivated.
So there are some keywords. Transcription factors, epigenetic mechanisms like DNA methylation, histone modification, and ncRNAs. Okay, let's read what it says here, and if you remember something, because really you do not need to go into detail. You're going to have questions about this. You just need to mention these processes, okay? But it needs to make sense. It says here, for example, during the process of differentiation, some parts of the chromosomes undergo supercoiling. This is around the histone, to prevent certain genes being transcribed. So one way of switching off a gene is supercoiling, which means that it's tightly coiled around the histone, therefore it cannot be transcribed. Okay? So that's one way of switching it off. So other areas uncoil, opening them for transcription so that new proteins are made. So that's switching on. So uncoiling of the DNA. Some genes are activated and others are silenced. As development progresses, more genes are silenced in each cell. Remember, the DNA of your muscle contains the genes, the same genes that the neurons in your brain. But the genes that make the neuron a neuron and that help it carry out its function in the muscle are switched off, and in the brain are switched on. And that can happen through supercoiling, the switching off. Okay?
You said about the silent, remind me, something that confused me was the heterochromatin and the euchromatin.
Mm-hmm. Okay.
Something, the silent condensed chromatin and the active chromatin.
The silenced chromatin would be the supercoiled one.
Okay.
Because remember, what is chromatin? Chromatin is DNA.
Yeah.
Chromatin is the very, very long chromosome, but there is still supercoiling.
Yeah.
Okay?
So you would say that the silent one is
It's the supercoiled.
Okay.
Silent meaning that the gene is there, but it's switched off.
Okay.
And this is important. As development progresses, more genes are silenced in each cell. How can you explain that?
Can you repeat?
As development progresses, more genes are silenced in each cell. What does that mean?
So they become more activated.
Are silenced.
Silenced.
Why do you need more genes to be silenced in each cell as development progresses?
So that you can narrow it down to like one gene.
So you can narrow it down not only to one gene, but to the genes that have to do with the specialization of a particular cell.
Mm-hmm.
So you don't want a muscle cell having genes activated that have to do with the brain cell. And you don't want the brain cell to have the genes activated that have to do with the muscle cell function. So as the cell becomes more and more differentiated, the genes that are not relevant to the function of that cell need to be switched off. Okay? It is a combination of the particular genes that are activated or silenced that result in the different characteristics or fully differentiated mature cells. Okay? So it's more of the same. So if you understand that in a differentiated cell, you're going to have only a specific group of genes activated that have to do with the function, and they're going to have all the other genes are going to be silenced. That's the basic way of saying it. Okay? Now there is a let's do this last bit, and then we answer the. You're gonna have to do the regulations and the therapy on your own, but I think that's more ethical. You can just read that. But let's have a look at this one because they talk about histone acetylation, DNA methylation, histone methylation, non-coding RNA, and transcription factors. So this is page 211. Maybe you want to have a look at that. So for example, here, they talk about hemoglobin. So this is a good example. Hemoglobin is it's a protein. It's made of 4 polypeptide chains, and it carries oxygen around the body. Adult human hemoglobin contains 2 alpha and 2 beta globin chains. Those are the polypeptide chains. Fetal hemoglobin has a stronger affinity for oxygen. Remember, the fetus is surrounded by water, so there is no breathing. So it needs to get the oxygen through the placenta from the mother, so the hemoglobin in the fetus is different. Okay? During human development, from embryo to fetus to baby, different versions of the globin genes are switched on and off. The levels of the different types of globin chain in the blood change through the 40 weeks of pregnancy and after the baby is born. Genes for the different proteins get switched on and off and are activated in different tissues as development progresses. Remember, hemoglobin is in the blood is circulating around the whole body of the baby. Globin production moves from the yolk sac in the embryo to the liver in the fetus and then to the spleen. This is interesting. Okay. Finally, at the time of birth, the genes in the bone marrow have taken over this function almost completely, and it's what is happening in us. Okay. Remember, your red blood cells die every 120 days, so we are constantly making new blood cells. The genes, so the cells in the bone marrow are stem cells, and they are pluripotent, using the terms that we've been looking at. So the genes controlling the production of alpha globin are needed in both the fetus and the mature baby. The genes controlling fetal gamma globin are very important during fetal development. This is something that maybe you don't have to memorize, but it's a good case to understand because you can have questions that are similar. So what you need to bear in mind here is that there are different polypeptide chains, and that by changing a little bit the polypeptide chain, you're going to change the hemoglobin affinity for oxygen. Okay? The adult beta globin genes need to be activated just before birth in the bone marrow. This is a clear example of epigenetics working in several tissues to change which proteins are made. The mechanism which controls the genes, okay, is the subject of much active research. They don't know everything yet, but they have discovered quite a bit. But we can work out how to reactivate fetal hemoglobin in children and adults. Okay, and they may be able to overcome problems that are present in genetic conditions like sickle cell anemia, thalassemia, which affect the structure of adult hemoglobin. So there are these 5 epigenetic control mechanisms. Okay, they are on page 211 that you need to know about. Histone acetylation activates gamma globin gene in the fetus. What is histone acetylation? It's a change in the histone, and remember the histone is the protein around which the DNA is coiled. Okay? So because it activates a gene, it means the acetylation uncoils the DNA from the histone. So it switches it on. Then, DNA methylation. The methyl group is the group CH3. Okay? When the CH3 attaches to the DNA, it silences, in this case, the fetal gamma globin genes. So methylation can silence some genes. And methylation is just adding the CH3 group.
So it's supercoiling the histone?
It's no.
Because it's silencing, so it's like deactivating it?
No, sometimes it acts as a blockage for transcription. It blocks transcription. Not everything is done through supercoiling. And then histone methylation appears to complement DNA methylation in silencing the fetal gamma globulin.
So the histone acetylation uncoils the DNA from the histone, and the methylation acts as a blockage for transcription. Oh,
okay. So it makes a little bit more sense. Okay, there is also histone methylation, but again, you see, they say it appears to do this, it appears to do that. They are not absolutely sure of all the details on how is it happening, but it has to do a lot with switching on and off the gene. Then let's look at this ncRNA means non-coding RNAs. So there are non-coding RNAs that have been associated with the process, but scientists are not yet sure what they do. So they find some bits and pieces of RNA that they are not really going to go out on the ribosome for protein translation. And they are thinking that it has to do with switching on and off the genes. Okay? Mm-hmm. So as I told you, you just need to know these 5 ways in which genes are switched on and off because it has to do with development, specialization. Okay. And then finally, the transcription factors. They are important to switch the production of beta globin in the spleen and bone marrow. Okay? Without needing to learn the definition of transcription factors and so on. So I'm going to stop here so we have time to answer 7D, the last bit, in the handout that I gave you. Because the one with the chromosomes is not going to be in the test. So let's do the last bit. So now I want you to answer that. And then we look at the answer.
Oh, this was literally a quiz.
Yeah,
All
that was
the
a quiz.
It was the full quiz.
That's actually
You see, next time you come have a lesson. Yeah, but you were not well or something. Yeah, you were not. It was after Cacopetria.
Yeah. Oh yeah,
I got COVID.
I think it's I hope so. I'm gonna look on that side. I'm gonna look on this side.
So it's 3 marks.
Yeah, no, 1 second. We're just doing the questions.
Mm-hmm.
So
It's 4:30.
She's bang on the top.
I can give you the answer if you want to take a photo. If you have to go.
I can write it down.
Mm-hmm.
So do we just write them in points?
Yeah, well, don't do that like Mano was doing last year. He was answering with a mark scheme.
But that's what she does for us this year.
Yeah.
This idea of we're supposed to know how to do it.
But that's
No, you are going to get the points, but
It's not explained properly.
Mm-hmm.